But I'm also always glad to talk about the research we do in our lab to a new audience. So it was very exciting. I have to say that also I became a little bit apprehensive in the recent days because I didn't know what to present, right? Because I usually present the work we do to our peers, and today will be slightly different. But I decided to still go through our research. I made a slightly longer introduction. Well, we'll cover some of the basics that you'll need to understand the last portion of the talk. So today we are going to talk a little bit about human brain development and evolution and try to answer at least a little bit to this question of what makes the human brain unique. So humans have evolved unique cognitive abilities. There are several of them. Here I listed just a few. We have productive language that allow us to communicate like we are doing just now, right? I'm speaking, and you are hopefully understanding everything I say. So that's a good skill to have. So we also have evolved theory of mind, and this is our ability to understand what other people are feeling and what other people, how are other people behaving because of the way they feel. And that's very unique if you think about it, right? Even now, you can look at a person and try to ascribe them a specific behavior or a specific way of interacting with others. We also have abstract reasoning, right? That I don't need to explain too much about this one, but you know how to make a deductive reasoning. You know, start from 1 + 1 = 2. That's a very good example of an abstract reasoning. Also, we are very good at social learning. We also developed this unique ability of producing and enjoying art. We also developed very strong moral reasoning, which is encoded in our legal system, right, in the law. And also, and this is probably one of the most important of all, we have cumulative culture, right? Because if you think about it, language is important, but what really changed our ability to develop a lot of things that we now consider normal is the appearance of written language, not just language, because a culture that doesn't write their thoughts doesn't accumulate knowledge, right? You only have, in each generation, you need to learn everything again, because it's not in writing. So when people started write things they know, then you don't need to do those things again. You already know they are likely true, and so you start accumulating. And so these are all unique things. And here you are looking at the brain scanned from side. Imagine you are looking at to me from my side, from one place to the other, and this is a human brain. We'll talk a little bit about these structures. In parallel with developing these unique abilities, we also became vulnerable to several psychiatric disorders, and many of them are unique to the human brain. And unique here is with an asterisk, because they are really not unique. They are unique because we are humans, and we described this disease based on our brain. And in that sense, of course, they are unique to us, right? But the funny thing is, we now know a lot of genes that are involved in several psychiatric disorders. And when you disrupt the genes the same way that they are disrupted in us when we have the disorder, and you disrupt them in animals, such as mouse or macaques, etc., you also alter their behavior, meaning that those genes underlie a lot of the development of the behavioral repertoire of the species. So in that sense, they are unique, but not completely unique. And so you probably know a lot of these, right? You have schizophrenia, depression, bulimia, autism spectrum disorder, but also bipolar disorder, schizoaffective disorder, factitious disorders, anorexia nervosa, several more. And of course, these are really serious disorders, at least some of them more than others, but they all affect tremendously the quality of life of patients and their families. And so there's a lot of focus on research on this, but of course, to understand these, we need to understand how the brain is built, right? And so that then we can understand what went wrong at some point. Before we go on, I just want to give a brief overview of human brain anatomy. Of course, this is one slide of 1,000-page book, but I'll try to summarize the best I can. So here you can look at the human brain. You have here the cerebellum in the back, and then the spinal cord, right, going through your spine that connects to your muscles for movement. And so when you do a cut like this, right, you are basically looking at me and you see a cut like this. This is what you get. And there's the famous gray matter, white matter. Most of the neurons are presented here in the gray matter. So this is the cortex, this sheet of neurons that cover the brain. And we'll be talking about the cortex quite a lot. There are other structures, but this is white matter because these neurons send their processes called axons, and I'll talk in the next slide, I'll explain you all of this, but the neurons are here and they send these long axons to connect to other parts of the brain and of the body. And they are involved in by one substance called myelin. It's a fat and makes this part white, basically. And there are several disorders that affect this myelin, for example, multiple sclerosis, etc. But it's not the focus of the talk today. If you cut the brain now on your side, when you are looking to me, if you see my brain in several sections like this is how it looks. Again, this is the cortex that we are going to be talking about quite a lot. Here you have the cerebellum. Here, it's an interesting structure called corpus callosum, which is exactly the same thing you see here in another view. And this connects the two brain hemispheres. And there are actually diseases where a surgeon will need to cut the corpus callosum to solve some of the problems, and those patients stop having communication between left and right hemispheres, and a lot of interesting problems arise. It's not the worst disease in the world, to be honest, but when you lose the left-right connections, it affects some of the cognitive processing. And here is the same thing, but now viewed from the top. Again, we are going to talk a lot about the cortex because the neocortex, so this structure here that you see lining up the brain and you see covering the entire brain, it's actually evolutionarily very recent. Only mammals have this structure. For example, reptiles don't have it, fish don't have it. Only mammals have this, and they are uniquely involved in a lot of things you consider to be present in the brain. For example, you have the primary motor cortex. This is where neurons in this structure send a lot of information. It's very complicated, but at the end it makes you move, right? Next to it, for example, you have the primary somatosensory cortex, which is a cortical area that it does the opposite. It senses touch, pain, temperature, etc. At the end, at the back of the cortex, you have the primary visual cortex, which processes information from our eyes. The retina sends indirectly the axons from the retina go there. There's a mid-structure involved. I'm not going to talk about it, but it does visual processing. And you have all of these areas, right? And then the prefrontal cortex is involved in a lot of things that are unique in human, or very specialized in human. And damages in the prefrontal cortex actually have shown to change completely the personality of a person, for example. There's a very famous case, a worker in the mines called Gage. He was working in the mine. There was an explosion. A bar of iron went through his skull, entered here, exited there. Didn't die. He was very lucky. Didn't cut any major artery or vein, so it just cut through his prefrontal cortex right there. Changed completely his personality. Became very aggressive, not the nicest person in the world. And that's when we started to realize that this is really a part of the brain that is very important for personality, but now we know it's very important for long-term planning, very important for top-down inhibition. You know, when you have, you want to do something, but it's like, oh, probably I shouldn't. You need to thank your prefrontal cortex. It's one of the last parts of the brain to fully develop. Actually, it continues to be myelinated up to your 30s. And that's one of the reasons, for example, why you have legal ages for a lot of things, because you want the prefrontal cortex to be fully developed and say, no, don't do that, right? What's inside this structure, right? And that's what we are going to talk about today. So there are several different cell types in the brain, and this is just a very brief summary of all the cells we have in the brain. So the first distinction there is you have neurons and other cells. So not all the cells in the brain are neurons. In this slide, I'm going to show you all the neuron types that we have in the brain. They can be divided in two main types. They are excitatory neurons or inhibitory neurons. The excitatory neurons are probably the most important cell type in your brain. So this is how they look like. They are called pyramidal neurons because you see here their cell body looks like a pyramid, basically. That's the only reason. And I said that they are the main cell in the brain because on top of being called pyramidal neurons or projection neurons, they're sometimes called the principal neurons. So they are really, really important. Because what do they do? So this is how a neuron looks like. You have a cell body, then you have this huge projection from it. This is called a dendrite. And this is where you receive, all these are dendrites, these black structures. It's where you receive inputs from other neurons in the brain. So you see these small structures here. These are called spines. And in this spine, you will have an axon, which is here. This red part will connect there. And this is how you form a circuit. And circuits basically brain circuits control everything you do. So you have the dendrites and the axon. The dendrites receive information, the axon projects the information outside of the neuron to another neuron. And they are excitatory, as I told you. So they excite the cell they connect to. If they fire, once the connection goes to the other neuron, it will make them fire as well. Of course, it's a lot more complicated than this, but this is basically the basis of the circuitry. Now, the inhibitory neurons do the opposite. They also connect to each other, and they connect a lot to the excitatory neurons. So sometimes, or most of the times, these inhibitory neurons send their axons, and their axons will form synapses into these neurons. So they connect to this neuron. And in this case, when they release their neurotransmitter, it will inhibit. So they will tell this neuron, don't fire. Now, any of you have any idea how many synapses, so how many connections one neuron in the human brain, in the cortex has? Any guess? 8,000. So each of the neurons receive 8,000 inputs. But now the cool thing about the brain is the output is always the same. Once a neuron receives all the information from all the other places into these 8,000, it will fire or not fire. It's completely binary. It will not fire more, fire less. It either fires or not. So there's lots of computation going on. Now, You can see that not all these neurons look the same. In the cortex, they are organized into 6 layers. And it's basically the same throughout the entire cortex. All the areas I was telling you have this organization. You have layer 1. Layer 1 doesn't have basically almost any neurons. It's only axons going, and it's important for integration of lots of signals, but doesn't have cells. Then you have layers 2 and 3. These are called upper layer neurons because they are up. If you look here, they would be closer to the surface. And these neurons are very expanded. The number of these neurons is tremendously expanded in primates and in humans. And they are important and peculiar because all their axons, so all their output, goes to other parts of the cortex. So this is how your visual cortex talks to your motor cortex, how your auditory cortex talks to your visual cortex, etc. These neurons in the upper layer send information across all these areas. Now, the layers 5 and 6 are very different. First, they are much larger. Look at the size of these. And then they project outside the cortex. For example, layer 5 neurons project to the spinal cord. This is a very famous circuit. It's corticospinal tract that makes you be able to do all of these, walk and touch your fingers, writing. All of these very precise skills come from these. These have the longest axons of the body. For example, you can imagine that a neuron in your layer 5 in the motor cortex sends an axon all the way, for example, to your lumbar spinal cord to move the legs, for example. And so those are very long. And now imagine a giraffe, for example. These neurons in a giraffe have an axon that goes meters all the way down to the spinal cord. So these are very, very big cells. And then layer 4 is very different because layer 4 is where the cortex receives all the input information. When I told you that the visual cortex, etc., receive information, for example, from the retina indirectly, because all your sensory input goes to a structure called the thalamus, and then the thalamus projects to the cortex this information. And it arrives in layer 4. There's only one sense, which is evolutionarily probably the oldest, that goes directly to the cortex, and that's olfaction. So olfaction doesn't have any relay, it goes directly to your cortex. All the others have a relay station. Then inhibitory neurons, there are several subtypes. And it's funny because in this case, the excitatory neurons are named by the layer they belong to. They are called layer 5 neurons, layer 6 neurons, layer 3 neurons, etc. The inhibitory neurons are named based on their morphology. For example, you have chandelier neurons because they look like chandeliers. You have basket cells because they look like baskets, so they really involve some of the excitatory neurons. And there are several of these cases. For example, this one is called bipolar because you have 2 poles and you are sending your processes for both. So this is a basic overview of the neurons, but there are also other cells, like I told you. And more research is being done in these cells. You have astrocytes, and there are several types of astrocytes, oligodendrocytes, and microglia. These are very important cells. The astrocytes are very important to support the neurons. They support them with energy, nutrients, they do the recycling of all these neurotransmitters, a lot of important jobs. The oligodendrocytes are the cells that ensheath the axons. So they wrap myelin around the axons, and they make so that the firing of these neurons is now much faster. It's like an insulated cable, basically. It insulates the axon so that the action potential propagates very fast. Microglia cell, this is the basic immune cell of the brain. So when you have an infection or when a cell is dying, it goes there, eats up the cell, recycles it, and it's all good to go. Hopefully, right? So these are, again, neurons and then glial cells. These are different, right? These don't have action potentials. They don't act on the neurons, make them firing or not firing. These are mostly supportive cells. I forgot to tell you an important thing. So the main difference between excitatory neurons and inhibitory neurons is on the neurotransmitter they use. So these are the molecules that the neurons release to one another. This was found several decades ago. So when I said that the axons connect to another neuron, they actually physically do not connect. They come very, 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 very close as you can imagine. You can only see actually in electron microscope. And once in these places of near contact, what happens is that the neuron upstream releases a molecule called a neurotransmitter that then propagates the signal to the other one. And the main neurotransmitter, excitatory neurotransmitter, is called glutamate, and the main inhibitor is called GABA. And so when GABA is released, it inhibits the neuron to fire. When glutamate is released, it excites the next neuron to fire. We are going to talk about other type of neurotransmitters that actually don't act like this. They just modulate. You all heard about neuroplasticity. So there are lots of other neurotransmitters called neuromodulators because they don't make the neurons to fire or not fire. They just modulate on how they fire. Now let's go to the real talk after this brief introduction. So humans have the largest brains among primates. And here you have an evolutionary tree with humans, right? Homo sapiens, our closest relatives, which are the chimpanzee and bonobos. Our last common ancestor was around 6 million years ago. So 6 million years ago in the savannas of Africa, these two species split. Then next to it we have the gorillas, which is the other African great ape. Then you have the orangutan, which is the Asian great ape. Then you have gibbons and several species of monkeys, including macaque, marmoset, etc. And you can see as you go from monkeys to apes, the brain grows, and as you go to African great apes, it grows even more. And then in humans, complete outlier. It grows quite a lot, right? The human brain is roughly 3 times the size of the chimpanzee brain. And so this could be the end of my talk, right? Humans have a larger brain, and that's what makes us unique, and that's it. But of course it's not, right? Because we know, for example, there are diseases, microcephaly and other types of diseases that which causes the patients to have smaller brains. And in a lot of these diseases, their cognitive abilities are basically very similar to an unaffected person. And so brain size probably doesn't explain everything. And we also know that people with larger brain size or larger heads are not necessarily more intelligent than people with smaller heads, right? So brain size probably doesn't explain the entire story. Although, if you have more neurons, theoretically you have more computational power, right? And so that's what I want to show you next is humans also have the largest number of neurons among primates. Here you have a graph where you have plotted the brain mass, so how much your brain weighs, and the number of neurons that are present in the cerebral cortex, so in the neocortex, that structure that covers everything. And you have several species here. For example, you have the hedgehogs. Here you have the rabbits and mice, tree shrews. You have the ungulates, hoofed animals, etc. And you can see that in all these species, there's a complete linear expansion. The larger the brain, the more neurons you have. But you can see that there are 2 groups that have more neurons than expected for the size of the brain. And those 2 groups are the primates, here in red, and the cetaceans, here in blue. So whales, dolphins, etc. And you can see that among the primates, humans have clearly the largest number of neurons in the cortex. But interestingly, the long-fin pilot whale, which is a species of dolphin, has more neurons even than humans have in the neocortex. So we are not the species with the largest number of neurons in the cortex. And I forgot to tell you, even here, an extinct human species, the Neanderthals, and you probably all heard about it, had larger brains than us, actually. And so that also tells you a little bit. We had smaller brains, but they got extinct. So I think we did, something right there. And so this probably, again, helps because, for example, elephants, which belongs to the Afrotherians, we know that elephants are very intelligent, right? Because they have large brains. So there is a little bit of a correlation of larger brain within your group and having higher cognitive abilities. But there's also more to it, like I told you. And one of those things is how the neurons are connected to each other. So brain circuitry, right? What connects to what and how. As you can imagine, that's very important. You need to have all those units for processing information, but you also need the right connections, right? If you put a computer with the wrong connection, it will not work. So you need the right connections for things to work. And in this case, I'm showing you differences in brain cognitivity in one of the most famous brain tracts. This in red is called the arcuate fasciculus. It's a long name. You don't need, of course, to know it. But it's very important because it connects all the cortical areas that are involved in language. So here in the prefrontal cortex, you have the Broca's area. So this is a cortical area that's involved in language production. So it has the name of Broca because it was the doctor who found that patients who had this part of the brain damage could not speak. They would understand everything you say, but then they cannot speak. They can produce sound, but it's all meaningless. They don't have a coherent sentence. And this is connected by this tract to this region that is called Wernicke region. And this is the opposite. This is a region involved in understanding language. So people have damage in this area. They don't understand anything you tell them. They cannot process language. But they actually can produce a sentence randomly. They don't have any problem in producing language. Of course, since they don't understand it, they also struggle quite a bit, as you can imagine. And why is this interesting? Look at the difference of this tract between human, chimpanzee, and macaque. You can see that in human, it's much more complicated. It's much more branched, especially here in the temporal cortex, where in the other species is completely absent, and in humans send all these projections down. Now, what is interesting about it is here in the inferior temporal cortex is where we do processing of what words mean. So you really need to have this complex branching to integrate all these language areas, and only humans have it. So I already told you about brain size, about number of neurons, and about brain connectivity. But there's even more to it, right? And now we are getting a hint of what makes us unique. It's not one single thing. It's accumulation of lots of smaller things, right? We have larger brains. We have more neurons. These neurons have different connections. And as I'm going to show you, these neurons themselves are specialized in humans. They have different molecular identity, so they are expressing different molecules that make them functionally slightly different. And when you accumulate all these things, you generate an enormous capacity of things that we have in terms of cognition and behavioral repertoire. So one slide that sums a little bit what I've been talking about. Here you have a human brain, a macaque brain. So this is a rhesus macaque, which is the most commonly used primate in research, and mouse, which is the most commonly used mammal. And they are at scale. We had to zoom in mouse, otherwise you would not see anything. So mice have a cortex that spans more or less 1.74 square centimeters. The human cortex is almost 1,100 times larger than the mouse cortex. Mice have around 4 million neurons in their cortex. Humans have 16 billion neurons in their cortex. So again, 4,000 times more neurons in the cortex. Um, Actually, does anybody know how many neurons we have in our brain total? 86 billion with a B. So that's quite a lot of neurons. This is just in the cortex, as I was telling you. Actually, the structure that has by far the most amount of neurons in the brain is the cerebellum. And so, again, more neurons, larger cortex. But this cortex, as I told you, can be parcellated into different functional areas. Again, now you can see what I was talking about. Here in blue, you have the primary motor cortex. In green, you have the primary sensory cortex. This is the somatosensory, again, for touch, pain, etc. This is the auditory cortex for hearing, and this is the visual cortex for seeing. But you can see that actually, across the species, these areas are more or less conserved. You always have the motor cortex in the front, followed by somatosensory, the auditory a little bit more ventral to it, and then the primary visual in the back. It makes sense that these areas would be very well conserved across species, right? Because all animals need to sense their environment and to react to it. They need these sensory areas and motor areas to react to it. Of course, they work in a very different way, especially the mouse. The mouse doesn't see very well, but has a very good somatosensory, actually. This is called the barrel cortex. Each whisker of the mouse has a specific cortical area map there. It's really amazing. And we don't, of course. We don't have any of that. We don't have whiskers. But half of our brain is used to process visual information because we are very visual animals. So recent studies have divided the cortex, again in functional areas, into around 180 areas. And you can see mice have around 50. So since we have more real estate, we can have more space for other functions that animals with a smaller brain cannot. To reach this level of complexity, because so far we have been only talking about adult brains, but to reach an adult brain, the nervous system has to undergo a very long developmental process. So this is how the brain looks from 23 weeks of gestation all the way to 19 years old. And from the time of conception until late adulthood, you have the cellular processes that will generate the brain. So one funny thing about the nervous system and the brain is that neurons cannot divide. So your skin is always dividing, right? You are always shedding cells, and you are being produced and replacing. Same with your intestine. Your entire digestive system is always being replaced. Not your brain, unfortunately, right? When you get all the neurons into place, that's all. That's all you get. After that, they will start dying, and nothing is replaced, because neurons cannot divide. Only the progenitor cells can divide, and you can see that it finishes roughly just before birth. So when you are born, actually, the younger you are, the more neurons you have, and the more synapses you have. It's interesting. And then as you age, it will go all downhill. But the neurons are produced in this ventricular zone. So these are the brain has these ventricles full of liquid called cerebrospinal fluid that goes all the way to your spinal cord. That's why when you do the spinal tap, you are checking the same fluid. That's why you do it, to see if there's something wrong in your nervous system. But lining these ventricles are the cells, progenitors, that will generate all the neurons that you have through this process called neurogenesis. Once the neurons are generated, they will migrate to their final place. Actually, let me show you just briefly. I think it's easier here. So this is the ventricle, right? This is a space full with liquid. And in embryonic and fetal development, these cells that line the ventricle is where the progenitors are. They divide, and then the neurons will go and migrate to their final place in the cortex, forming these layers. And once they arrive to their final place, they start extending their processes, their axons, their dendrites, and start connecting to each other, forming the circuitry that basically controls everything we do. And that's a process called synaptogenesis, formation of synapse, formation of point of contacts between neurons. Then their axons get wrapped up by myelin, and as I told you, it goes all the way to 30 years old in the prefrontal cortex, and you have also synaptic pruning. So it's the use it or lose it process. If you are born with a lot more synapses than you will be end up using, and the ones that are not used, you trim them out. And so basically myelination and synaptic pruning is what closes this period of neuroplasticity. That's why when you are young, you can learn everything very fast. When you are not young, it's very hard to learn new things because the system says, okay, I have all the information we got, there's no more place for new things. And as you can imagine, if these processes are always controlled by which genes are being expressed in each cell type, that is basically governing each of these processes. So it's very important to know what are the genes that are being expressed by all these cells that basically are essential to form these. And we know quite a lot about this. For example, if a gene disrupts the progenitors, it means that the progenitors will not be able to generate as many neurons as they usually do. And you usually have microcephaly. And today I'm not going to talk about it, but we did research on microcephaly when we had the Zika infection, the Zika virus infection. You remember it was roughly 10 years ago that a lot of babies were being born with microcephaly, and we actually detected and we found the mechanism that was causing the microcephaly. We found that when the virus selectively infected the progenitors, and these progenitors were not able to finish cell division and end up dying, meaning that neurons could not be produced, and so you had a much smaller brain, microcephaly. If genes that are disrupted are later in neurons, and especially in neurons and how they connect to each other, you'll have more psychiatric disorders. So the earlier the mutations are, the more structurally damaged your brain will be. It'll be either with microcephaly or even more severe abnormalities. But the later it is, when it affects the circuitry, you will have more psychiatric disorders. And we'll just touch a little bit on some of them. Now, this is a schematic for you to understand how this brain division occurs, because this is rather important, because it will explain how a bigger brain is formed. Imagine that this is time, right? From left to right, you see time. And these are the cells that are lining the ventricle. You imagine, these are the neuroprogenitor cells. They divide, and they generate a neuron, and the neuron will go to the cortex. Now, the cool thing is, you see that these cells, these progenitors, have this process extending all the way up. When these cells divide and generate a neuron, the neuron actually uses this fiber to migrate along and go to their final place. It's really remarkable. And it's an inside-out way of formation of the cortex. The first layers to be formed are actually layer 6. So the layer 6 neurons migrate and stop there. Then the layer 5 neurons need to go through the layer 6 and form the layer 5. The layer 4 needs to go through layer 6 and 5 and form layer 4, etc., etc. So it's an inside-out cortex. And this is basically the organization I showed you in the previous slide. You have the excitatory neurons in each of these layers being formed by this. And I actually I want to show you some drawings and some pictures of the process itself. So this is a section from a brain. The progenitors are lining the ventricles. These are where the cell bodies are. Progenitors, these are the processes, and you can see here neurons migrating through it. And this is a real brain. These small dots here are neurons migrating through these fibers of the progenitors. And this is a schematic basically showing this is a neuron. They wrap themselves around this process and they use it to migrate to their final place. So you could imagine one potential theory to explain a larger brain would be this one. So this is a mouse brain. This progenitors have two types of division. First, they start dividing symmetrically. And we say symmetrically because one cell divides and the two daughter cells are exactly the same as to each other and exactly the same as their mother cell. So this is, again, an horizontal expansion of the progenitors. And then they start dividing asymmetric, and each of them gives you a neuron and another progenitor cell, and so on and so forth. So you can imagine that if we prolong, this symmetric division, and we have then a lot of progenitor cells, each of them will be able to generate neurons, and so you can increase the number of neurons that you'll have in the brain. And actually this is called the radial union hypothesis, is very famous, saying that you increase the number of progenitors, and then each progenitor will send neurons to the cortex, and that's how you expand. If you have more progenitors, you'll have more neurons. Now we know it's not exactly like that. We know that these progenitors in humans also divide symmetrically first and asymmetrically last, and they generate neurons. But humans have this extra cell type. It's other type of progenitors, intermediate. So the progenitors generate other progenitors that will then generate the upper layer neurons. And this will create a tremendous expansion of the cortex. And just if you think about physically, it makes sense that in a more or less spherical structure, the more it grows, the more the upper layer needs to grow, right? Because it takes much more surface area in the outer parts than in the inner parts. And so this is what was found recently, that humans have these extra progenitor cells that expand tremendously the upper layer neurons, and so that's how you get all this expansion of the brain. So I already showed you this. This is, again, a brief summary of what I've been telling you. The organization of these cortical areas is conserved, but also variable across mammals, because I told you the primary areas, motor and sensory, are conserved. But one thing I didn't tell, because I wanted to say before this slide, is look at the amount of gray, white, and red surface in each of the brains. You can see that here the primary areas cover almost all of the mouse brain. But as we go to primates, monkeys, look at the amount of surface that is not covered by primary areas, and then in human, even more. These are called association areas, and they are very important because they integrate all this information, and as I told you, then you have specialized areas in the frontal and temporal cortex, for example, that are involved in language, in long-term planning, all of these things. Actually, long-term planning, I think, is one of the truly unique abilities of humans. Because since we have a good understanding of our lifespan, etc., we really make very long-term plans. Try to teach, your dog or cat to save for retirement. Or even to save food for later. They don't. They eat as much as they can immediately. It's very hard to do long-term planning, and we are actually very good at it. Because look at the expansion of the prefrontal cortex in human compared to the other species. Also, of course, these variations, as I was telling you, provide, of course, the basis for species-specific cognition and behavior, and this is all generated throughout development, and this process is affected in major diseases. Now I'm going to talk a little about our research, just a couple of studies. So since this prefrontal cortex is so important for humans, we really wanted to understand if there is anything different here. I mean, we already know it's larger, but is there anything inside that makes any difference between us and other species? And that's what we did. So we had post-mortem brains from all these species, from human, chimpanzee, macaque, and marmoset, all adult specimens. And what we did was to dissect the same cortical area in all of these species. This is called the dorsolateral prefrontal cortex, one of the most important cortical areas, if not the most important, for all the things that are involved in high functions. For example, cognitive abilities, working memory, long-term planning, all these things I was telling you, the dorsolateral prefrontal cortex is very involved. And so what we did, we dissected this brain region, and then we isolated single cells. It's complicated, I need to know how it's done, but so we isolated each of the cells inside this structure, and we characterized which genes were being expressed in each of these cells. And look at this. I'm not going to explain this, so don't worry. This is just to show you how many cell types we found. We actually found 114 different cell types in this cortical area in this species. That's why I told you when I was showing you that excitatory inhibitory news that this is a simplified version, because this is not the full version of it. But I want to tell you just a couple of things. So in magenta here, you have the excitatory neurons. And these are divided by to which layer they belong and to where they send their projections. Here in green, you have the inhibitory neurons. And they are characterized differently by, again, their morphology, how do they look like, and which molecules they express. Then you have the glial cells, and then you have other cell types, because we also have blood vessels, right? We have all these cells that are also present in your brain, but have nothing to do with the nervous system. They're circulatory system, vascular system. And so now that we had these, we actually had profiled 600,000 single cells. So almost, more than 500,000 cells. Roughly 150,000 per species. And so we wanted to compare because we wanted to know what is unique about the human brain. And we thought about 4 potential things that would be interesting to study. So when you are comparing across species in an evolutionary comparison, several things can happen. Maybe one of the species have a new cell type that is not present in the other species. And as you could imagine, this is rare. This is a rare event. Most of the animals have the same type of neurons, especially some that are so closely related, right? These are all primates. You might also have changes in subtype abundance, maybe, and I already told you this, humans have more upper layer neurons than the other species. So changes just how many you have increase, again, the computational power. You also can have different heterogeneity among the same cell type. For example, imagine that you have layer 3 excitatory neurons in human, chimpanzee, and macaque. But in macaque, they're all very similar. All the cells that are in this layer, they all look alike. They are almost all like twins. But then you look in human, and they're actually quite different, meaning that they probably have different functions, right? Because you assume if they're all the same, they probably work more or less the same way. If they are different, they probably have different functions. So we also checked that. And finally, for the exact same type, layer 3, again in human and in chimpanzee, what is the difference between them? What are the genes that are different? What's happening? And so I don't have time to talk about all of this, and I decided to focus on the most important thing for us that we are now doing quite a lot of research on. Focusing on the green cells, inhibitory neurons, right? Those are the neurons that release GABA and inhibit the excitatory neuron to start action potential. This is all the interneurons we have. Actually, in this map, each of these small dots was a single cell we profiled. And in this analysis, what this method does is it groups cells that are similar together. So in this map, if physically you are close to each other, you are probably very similar to each other. And that's how we classify cell types. I can see several names here. For example, basket, these are basket cells, ones I was telling you. Here, CHC, these are the chandelier cells I was telling you. So lots of cells, it's complicated to go through all of these. We are particularly interested about this bridge here. Why? Because we found that there is a difference there. So these cells, these interneurons, are characterized by the expression of this gene, TH. It's tyrosine hydroxylase. I will explain in the next slides what it does. But for now, let's call it TH. So these neurons express this gene TH, and they are abundant in human, macaque, not so much in marmoset, but in human and macaque, yes, but completely absent in chimpanzees. And that caught our attention immediately. This is just for you to see how the neurons look like in the brain. So we are able to label them and then to take an image in the microscope. This is how do they look like. So I wanted to tell you what TH does and why it's important. So TH is the gene that encodes this protein called tyrosine hydroxylase. And so what does this do? It transforms tyrosine, which is an amino acid, in L-DOPA. And a lot of you probably heard about L-DOPA because this is actually levodopa, the Parkinson's treatment molecule. And then DDC, it's another gene that encodes another enzyme that produces dopamine from L-DOPA. And so this is how you produce dopamine. Dopamine is a neuromodulator. As I told you, I was going to talk about one. This is it. And dopamine is very important. So it's very important for you to plan and control movement. And it's the main system that is affected in Parkinson's disease. So the dopaminergic cells that you have die, so you don't have the release of dopamine in the brain, and you don't have the ability, especially to start and stop movement. It's very, very hard. And so this caught our attention because, okay, we found this difference between human and other species. What's going on here? They are not supposed to be here, first place, because I wanted to have this schematic. Again, this is the brain seen by the side, from the side. This is where the dopaminergic cells, so the neurons that produce and release dopamine are. They are present in your midbrain, very low, and they project to your entire brain. And especially, this is the most affected one in Parkinson's. It's the ones that go from substantia nigra to the basal ganglia. It's absolutely essential for motor planning and coordination. But they also go, again, to the cortex, to a lot of places. Now, the interesting thing is, this system is so important that it's very, very, very conserved across species. If you look at the brain of a mouse, it has the exact same cells projecting to the exact same places, because mice also need to plan very well their movement, right? So very, very well conserved. But now, do we have some of these cells also in the cortex in human? Actually, we do, and that's what we have been doing recently. So these are, again, neurons, and we label them for TH. So we try to identify cells that express this. And you can see them in humans. They are completely absent in gorillas, chimpanzees, and bonobos, but they are also present in the other species, in orangutans, baboons, rhesus macaques, pigtail macaques. And now you could ask me, but Andre, you just said that they might be specific to humans, and now you are showing that they're present in all these species. Because, of course, it's more complicated. It's always more complicated, right? So in humans, we try to label these cells and see if they express TH and DDC. Remember, to make dopamine, you need two of them. Otherwise, you just make L-DOPA, which will not give you dopamine. So we found that in human, but not in the other species, these cells express TH and DDC, so they could produce dopamine. So we thought, oh, this is really cool. We found a new cell in the brain that actually produces something important. And so we continued to do these studies. And then now we used advantage of stem cells. So these are induced pluripotent stem cells. I don't know if you heard about them. So these are not embryonic stem cells. These are stem cells generated from adults. You can get cells from your skin, and from your skin, we can de-differentiate them into stem cells and then differentiate them into whatever cell type we want, or we can. We know how to, right? So we did that. We had in the lab iPS cells from donors. And so we decided, okay, let's make cortical neurons and see if we can generate TH neurons, because if they are in the brain, maybe in a dish we can also get them. And we were very happy to know that we can get them, so we could get these TH cells. Because for a neuron to be able to produce and release dopamine, again, it needs to release it. It needs to be able to transport it. You can't still produce dopamine and don't have the machinery in the cell to release it to the next neuron and therefore influence that. And that happens quite a lot in the brain. And so we had to know if the neurons in human could transport dopamine, so they are able to produce and release. And now that we had these cells in vitro, of course, we can do experiments. As you can imagine, we cannot do experiments in the human brain, right? So we always need to find, that's one of the caveats of my research that makes it a little bit harder than most research, is that I want to study the human brain, but you cannot do direct experiments in the human brain. So we need to find clever ways on how to circumvent this. And in this case, we use this assay, which is actually quite remarkable. So you have a fluorescent molecule here in green that is very, very, very similar to dopamine. But it's masked. It has a capsule around, and if you look under the microscope, you don't see anything because it's masked, right? There's no fluorescent. Now, the cool thing about this molecule is if it goes into the neuron, it gets unmasked, and the neurons become fluorescent. And so we can see if the neuron transports it or not, because if all the neurons are not if the neurons are not fluorescent green, they don't transport. But if they become fluorescent, they were able to pick it up, right, and become fluorescent. And so that's what we saw, and I was super excited. I remember this was probably very late at night. My wife would probably know better. And when I say late, probably like 2 or 3 a.m. in the lab, right? But I started seeing cells becoming fluorescent green, and of course I got super excited. And so at the end of the experiment, I check if these same cells express TH and ADC, and they did. So this means that they could produce dopamine, and they could also transport dopamine. Finally, we checked if there was any dopamine in the media, because again, this is in a dish. If the neurons are releasing dopamine, it will eventually go to the media in the dish. And so we did a chemical analysis, and we were able to find dopamine in the dish. And so this got me really, really excited, because now we have shown that humans have these unique dopaminergic cells in the cortex. And then I dig into the literature, and somebody had found these weird cells in the cortex. They didn't know that they were dopaminergic. They just checked for TH, like we had done before, all of these. And they checked these in Parkinson's patients, and they were all depleted as well. So not only the ones I showed you are depleted, but these ones are also depleted in Parkinson's. And so now we are doing quite a lot of research on this. This is just to show you that the entire machinery, right? You need to produce dopamine, so you need to have TH and DDC. You need to package dopamine into the synaptic vesicles, so you need these proteins, and you need to release it and uptake it, and you usually also have an autoreceptor for feedback control. And only humans have all this machinery. Monkeys have cells that have TH, but they don't have the rest of the machinery. So these cells are really unique to human. And so we kind of changed our understanding of the dopaminergic system, that before we thought it was like this, but now we know that humans also have these cells. We are now continuing to working on this, trying to identify what is the real function. And it's going on. We are using in vitro models. We are also using mouse models, etc., to see how having these extra cells can affect ER behavior. And also their influence in Parkinson's disease. And so, right on time, so it's like 1 hour. Of course, I want to acknowledge all the very talented people in my lab that did most of the experiments I showed you. Also our collaborators, both at UW Medicine, but also outside at Yale, also in Barcelona, and the National Chimpanzee Brain Resource, because I don't know if you know, but it's actually impossible and illegal to do research in great apes. So great apes have the exact same legal protections as humans. So we need to use post-mortem brains. And so this National Chimpanzee Brain Resource, what does is when a chimpanzee in the sanctuary or in zoos dies, they are able to collect their brains very fast, freeze it, so that then we can use for research, which is actually quite remarkable. Of course, also for the Wisconsin National Primate Center, and to NIH, the Brain Behavior Research Foundation, Simons Foundation, International Rett Syndrome Foundation, and Brain Research Foundation for funding the work we do in the lab. And thank you all for being here.
Thank you. Thank you. Thank you, Dr. Sousa. My head is full. Please raise your hand if you have a question. You don't know how to phrase it exactly, do you? You have lots of questions.
We got one here.
I'll try a question on this. Is there an intersection or relationship between neurological development and early childhood education, for instance, reading to a baby as soon as they're born, exposing them to music, exposing them to language and things? Is there?
So yes, the answer is yes. So children have very plastic brain, right? As I told you, you can teach a child how to speak probably 20 languages, and they will pick up. For example, I came to this country when I was already older. That's why I have this accent, and I will always have this accent. I can try the better I can, but I will always have an accent. If I would come when I'm 6 or 7, I would have no accent at all. And in that sense, it has to do with that plasticity window and the critical periods that I showed you in terms of myelination and synaptogenesis. So, but myelination might be even the most important factor. And past that period, you lose a lot of plasticity. So that's why you should expose your children to as many things as possible, of course, without overwhelming them, because then you'll also do a disservice, right? But they are really like sponges, right? They can learn everything very, very, very fast.
Over here.
Yeah.
I hope this is not irrelevant. What is your understanding based on today's knowledge of the blood-brain barrier to molecules? I'm thinking of particularly, for example, hydroxyurea. You probably are acquainted with that for the treatment of cancers.
Mm-hmm.
And so is it crossing the so-called blood-brain barrier?
So,
or is that a bad concept these days?
Yeah. So that's something I didn't talk at all here. So the blood-brain barrier, it's like the name says, a barrier that basically works as a gatekeeper for things not getting into your brain. So your brain is probably your more precious organ, right? And so it has a lot of protections. Look at our skull, right? You don't have any other organ so well protected from injuries and insults from the outside. And even in terms of vasculature, you have this structure called blood-brain barrier, which is very complex molecularly and cellularly, that basically doesn't allow a lot of chemical and substances to reach your brain. They are in circulation, but they never go to astrocytes, neurons, etc. And as always with our systems, sometimes they are too good for our own good, right? Because for treatments, for example, there's lots of substances that cannot cross the blood-brain barrier, and we would love that they could, because then you could actually activate a lot of things. But again, think a bit like your immune system, because it's kind of similar in that sense. Your immune system is very good at reacting to any foreign agent, right? Any pathogen, anything. But sometimes it's also too good. It infects, like autoimmune diseases, right? Suddenly it recognizes a protein in your cells as a foreign pathogen, and it starts attacking your cells, and it kills it. And it's very hard to change these innate functions of the brain, of the system. Now, for the brain barrier, blood barrier, one interesting thing is we now know some substances that can make it leakier temporarily. So we can basically make it more permissive for a short period of time to allow things to go in, and then remove that and close it again. The problem we have is usually when we need to do this, it's to already patients that have lots of problems, right? And when you open, then lots of things can go in, not only the molecule you want. And so people are still working on special chemical structures that basically will encapsulate whatever you want to give, so that maybe if we are permissive in this way, let this thing go, but not other things, you'll probably be able to have better treatments. So it's a promising field, but we are not there yet, unfortunately. Yeah. But again, as everything in research, I can say this: we are not there yet, and next week a new study will come, that will show that it's possible. That's the beauty of science.
You talked about how when you get older, sometimes it's harder to like when you're younger, you learn more and stuff. So how does it work when somebody gets hurt and then they have to learn to like read or talk again? Like, how does the brain go through that?
It's very similar. Again, the older you are, the harder it gets in general. But there are actually some very interesting research. So, for example, cases of stroke, right? Patients have a stroke and, for example, and has aphasia afterwards, cannot speak, loses the ability to communicate. Again, it's the older you are, the harder it is to recover from those. But there's actually a very interesting approach being done in a lab in Boston, where they actually use music to retrain your ability to language. It's almost like a hack of your brain. You didn't lose the ability to process music. It's just words, but the sounds you can process them. And so, with a lot of physical therapy and musical therapy, they start using your ability to process music and use that processing to start processing language with the same system, associating words with sounds. And it's actually remarkable because it works. Of course, you don't regain 100% of capability, but you do get to talk again, which is, again, remarkable, I think. But again, the older you are, the harder it will get. But you are still young, so.
Okay, Andre, you were talking about Parkinson's.
Mm-hmm.
Now, is the same principles also available for those with RLS, or restless leg syndrome?
Yeah, so they are slightly different, not slightly, quite different diseases, right? Parkinson is more aggressive. So there are a lot of diseases that affect motor ability that are not related with dopamine. That's the first thing. For example, Huntington disease, right? It gives you ataxia, and it's really severe disease. It affects also the basal ganglia, but it has nothing to do with dopamine. We don't know, and we think, from what I know about them, I don't think these TH cells that we found, so these dopaminergic cortical cells, are involved in any way with tremors or anything like that. What we think happens here is this. We also don't think it's involved in Parkinson's, probably at all. Well, slightly. Let me explain. So the current theory, what we think is the production of dopamine is very costly to the brain. So I will need to go a little bit into detail. So the production of dopamine and all these neuromodulatory molecules creates a lot of oxygen species. And oxygen is very toxic for the cells, and it ends up damaging the cells long term. So these cells are very vulnerable. And what we think happened is this. So you remember when I showed you the human brain and the mouse brain, right? Mouse brain, very small. Human brain, huge. The dopaminergic cells in the midbrain, that slide that I showed that was conserved across all species. it increased in size almost nothing from mouse to human. Of course, it's larger, but the rest of the human brain increased a lot, and now you have a very small number of cells that needs to be sending dopamine to all these brain areas, and so it needs to produce a lot of dopamine and be always firing. That's one of the factors that we think makes our brain more vulnerable, because these cells, if we had more of them, if it had grew as much as the rest of the brain, we would have way more to send to the rest of the brain. And what we think now is that what creates the vulnerability is this production of dopamine. And that's why both the midbrain cells and the cortical cells die, even though they are completely different. So the physiology, the way they work is really completely different. And so we think it's uncoupled. What we think happens is evolutionarily, these cells gave us an evolutionary advantage, because now we have local dopamine being also released that doesn't rely so much in these long projections. But we also found these dopaminergic cells in the striatum. We are now trying to work on them because they are a little bit harder to study, but now we have more tools, and we are trying to see if in the striatum they have the same. So basal ganglia, they have the same molecular properties as in the cortex. If they do, then they probably could be important for any tremor disease, and probably we can try to target them. If not, probably not.
We probably just have time for one more question. Is there someone else who has a question?
I think
What happens to the brain during addiction?
Oh, that's a very good question. So, it's good that you talk about it, because I completely forgot to talk about it. So dopamine is completely involved in addiction, actually. Again, in that schematic, you have the substantia nigra dopaminergic cells projecting to the dorsal basal ganglia, and then you have the ventral tegmental area projecting to the nucleus accumbens. And this is actually the main circuitry that is involved in addiction. And addiction has all to do with dopamine. So dopamine has a lot of functions. One of them is in the reward system. So, for example, it's known that when people gamble, for example, or when you are drug users, before the use of drug or gambling or any addictive behavior, you have a peak of dopamine release. So dopamine is very important, and it's now very studied in this new field called neuroeconomics. It's basically how your brain judges the cost benefit of any action. And so in this case, it's known that people who have, a potential rewarding situation, you have a dopamine peak. And then you either satisfy the condition, so the higher the dopamine peak is, the higher the reward needs to be to satisfy your prediction, right? And so usually in addiction, it starts like this. It starts a very small dopamine peak, you get the reward, you're happy. Next time you do the same behavior, your dopamine peak will be slightly higher. So you need a little bit higher reward to satisfy the prediction. And you can see where this is going, right? Dopamine peak starts to be higher and higher and higher, and you need to have more higher and higher and higher reward. And this is what will create the addiction, because if you don't get the reward, you get all the behaviors that are typical from a person with addiction who didn't get the reward, right? And so you create this loop that it's very hard to break.
Thank you.
Yeah, thank you.